Okay, everybody. I was trying to get that set up to push to YouTube, but it looks like there's something going on. So I'm just recording it through Zoom. All of that should be working now, I think. Let me open this up. This is we left off here. This is right where we're at. I want to back it up right to this because after when we lectured on this, and I stopped uh, Friday. It kind of left me thinking a little bit about it. It was actually last night when I was kind of thinking a little bit about it, and it raised a couple of questions. I don't actually know exactly all of the details about the study and how many people were questioned in it in the first place, but then it started to make me wonder, what if the situation were a little different? What if there were the opportunity where everybody was on the same page and we had universal health care where everybody was afforded the same options? What sort of potentials might we see in this figure? What sort of trends could we potentially see? Do you think it would look more like the blue guys, the red guys even out, be equal, be different? What sort of potentials do you think could be there? And then I thought, you know what? I ought to make this some extra credit. So I will get a formal posting on, I'll post it on the Moodle after lecture here in a bit. But I would like you to address each of the three bits right on here and comment, speculate. There is no right or wrong answer to me. I'm just curious what you think might be the opportunity if everyone were on a level playing field. What sort of impressions do you think we might have with receiving antibiotics or not in each of these? And I'm not going to go through and say that. You can look through it and then think about, and I will see if I can make all of this post all together. So I'll have the details, the specifics. I just want, a few paragraphs. I don't want anything too lengthy. I don't want 2 sentences. But give me your take on this, addressing each of these, and I'll make it due right before this test, which I want to say, if I remember correct, is the 19th. So we'll make it due on the Thursday before, on the 18th. And again, I'll have more specifics spelled out on there because I just thought of it. So give me a second. All right. Any questions or concerns before I get into this chapter, to the real deal? Okay, so I will tell you, it's good to be back. Not that it wasn't nice lecturing from my house, but it is good to get out of the house and actually be out. That's good. So we left off thinking a little bit last week, thinking a little bit about antimicrobials and some of their potentials. And this figure is one that I think is good to think about. It's really the bold stuff. The individual drug, and we're going to have a handful of slides here in a second that kind of show the actions of some of these, but really it's these different types of classes of antibiotics that I think should be more of a focus. Trying to memorize the respective names that you'll get introduced to as we move throughout is not really necessary for this, but a little bit of the mode of action. How is it that all of the varied classes of antibiotics target specific bad guys, if you will? Oops. So starting right off, because that's where my pointer was, initially there are cell wall inhibitors. These, when we start thinking antibiotics, we oftentimes think about all sorts of different types of drugs, and those that are more specific toward the foreign invader, kind of the better off you are. So in this circumstance, when we target something with a cell wall, that's good because our cells don't have cell walls. So the action on these antimicrobials has very little ill effects on yourself, if you will. All right. So cell wall inhibitors, and there's a handful of different potentials. We shifted over to the plasma membrane. That's maybe more of an issue because we also have membrane. All right. But there are some definite differences as far as some of the classes and actions with this. But potential for maybe, sometimes you take some of these antimicrobials and it makes you feel a little ill, and it could be because it's also targeting your own specific cells as well. Maybe not as much. Targeting the ribosomes, bacterial ribosomes are different as far as size-wise. So trying to target them, but there are still issues where it may end up impacting your own cells. And thinking about it this way, when we think about our mitochondria, they've got bacterial-sized ribosomes. So taking antimicrobials that are targeting the ribosome do have the potential to impact your cells. Metabolic pathways like folic acid synthesis is a big one when we think about this. That impacts the bacteria. We get folic acid through our diet. So targeting the bacteria can synthesize it on their own. Targeting something like this truly is specific against those guys. And then DNA or RNA, dependent upon the types, impacting their synthesis. So replication or protein production, one way or the other. And these are, again, thinking mechanisms for bacteria. There are definitely some similarities when we think about antifungal agents trying to target some of them. We'll see some similar, at least a few similar approaches. There are definitely more of an issue when we start shifting gears and thinking about eukaryotic pathogens, because our cells are eukaryotic as well. So the targets are definitely a little bit different to ensure that you're not causing some serious harm on yourself. So like I said, you don't need to get bogged down with all of the specific names of antimicrobials. But real quickly, as we introduce a little of it, penicillin and a class of derivatives, they all have this ring structure, as you see here. And then the R group, these side chains, the stuff poking out, makes them a little bit different, right? So these are what we call, this ring structure is what we call a beta-lactam ring. And it really is, again, the kind of the backbone of these classes of drugs. So they've got, as you see here, specific activities, gram positives, a lot, and there's a few that target negatives as well. Kind of broad spectrum, if you will, with that. There are, and we will see as we get toward the tail end of this, different mechanisms for resistance to try to avoid some of these specific agents. And some organisms have an enzyme called beta-lactamase. Beta-lactamase has the ability to target this ring structure and basically split it in half, allowing that microbe that's got that enzyme the ability to chew it up and to metabolize it, to break it down. So in those types of organisms, we would classify them as being resistant to these types of drugs because they can degrade the molecule itself. And there's mechanisms trying to target some of that resistance through biochemically altering some of those structures a little bit, trying to make it so the enzymes can't target it. This next one, these are protein synthesizing, inhibiting antibacterials. So a couple of different types, and basically targeting some portion of the ribosomes, right? So 30 or 50S, and you can see where they're coming in, impacting it so that either the messenger RNA is not read, so you can't bring in the proper amino acids, or targeting it somewhere within there as the tRNAs are trying to move through, blocking the whole process one way or another. So again, different mechanisms, but the end goal is to inhibit the protein production in the first place. The antimetabolites, the sulfonamides, trimethoprim, these guys act by blocking the folic acid synthesis as they basically drive a wedge in that. It prevents the bacteria from producing specific nucleotides. The purine and pyrimidine biosynthesis is halted. And if you can't replicate your nucleic acids, you can't replicate. Shifting a little bit, when we think about fungi, they are definitely a little bit more of an issue when we think about trying to treat them. And we don't use the same antibiotics that you would think about treating bacterial infections. You don't give a dose of penicillin and think that's going to cure up a yeast infection. It's not the same, all right? So we need to think about other strategies, other mechanisms. And as we showed some of that with the classes for bacterial antimicrobials, when we think about some of that, well, those cells are different, but trying to inhibit protein synthesis with the fungi, maybe that's more of a problem because it's going to cause the same issues with our own cells, right? So one mechanism is with in the plasma membrane in cells. We see more than just phospholipids. There's some cholesterol molecules within our own cells that lend to a little bit of the fluidity of these cells. And the fungi have a kind of a cousin, if you will. Ergosterol is a cholesterol-like molecule. It's got a similar ring structure, some varied side chain coming off of it, but a close cousin, if you will, all right? This is found in the cell membrane of the fungi, and with that, we can have some antifungal agents that are targeting ergosterol or ergosterol synthesis, and that is something that's different than our own, and it can impact them respectively. There are some potential issues with some of those agents where they do have a slight impact on the cholesterol within our membranes. And not all cholesterol is bad news in the first place. So this image showing a few different ideas for the antifungal targets. And there are a couple of different potentials as far as how this might be effective. With that said, and again, it kind of depends on the type of fungus, but with that said, oftentimes treatment with antifungal agents is something that takes a considerable amount of time, right? Weeks, months, more likely, maybe prolonged for upwards of close to a year with some types of fungal infections. That sets the stage for some issues and definitely ill effects as a result of these agents, right? So definitely some side effects. So there are potentials where we may inhibit DNA or RNA synthesis, the impacting the mitochondria or microtubule functions within the thinking replication and the synthesis of those fibers to separate out the strands of the sister chromatids as it's been replicated. We can look at more issues as we move in here with some of the ergosterol molecules, as we said, targeting that. So inhibiting the synthesis of it, disrupting the membrane with some of these Pauling drugs that causes permeability through that membrane. And then out on the outside, we can see chitin and inhibiting the synthesis. This is more cell wall type of material, the chitin, big long polysaccharide chains. And then the synthesis of the beta glucans, also a potential target. So a handful of different classes of antifungals targeting, and this is maybe more as we look at the membrane and cell wall, more of the approach that is utilized for these guys. So this image is type of fungus, Pneumocystis, Pneumocystis jervecii, and this is a sputum specimen. So these yeast cells, you can see the dark border around them, very thick chitinous cell wall that these cells have. Here's a good shot with the nucleus of these little yeast cells. And these are an infection that's oftentimes found in individuals with HIV or that have progressed to the stage of AIDS. Usually that's where we start seeing all of these issues when you've progressed to that state. Numerous types of infections, and pneumocystis is one that is kind of a red flag that's raised. You kind of think there's some serious issues, because normally you don't typically think of these colonizing unless you've got some serious impacted immunity in the first place. So that's kind of a good segue to shift over and start thinking about some of the antiviral actions. And there, when we think about this, it's, although it looks like there's a handful of different potentials, not a tremendous amount really compared to some of the other mechanisms. And part of that is because, you know, a virus is basically bare bones in the first place. So we think about a few things with this, trying to impact the nucleic acids, DNA or RNA, whatever these guys are made of, trying to target that, or trying to target this production of their capsids, their shells, if you will. So a few mechanisms to try to inhibit the synthesis or the replication of their nucleic acids, whatever it might be. So acyclovir is an analog to guanosine, and so as it gets in there, it impacts the DNA replication where guanines should be coming in. It impacts that. The antiretroviral therapy, there are a handful of different mechanisms. As you can see in here, we've got little X's where we're inhibiting specific points of action with these virus. So HIV, it could be blocked as it's trying to dock or recognize the specific cell. Those receptors on the cells have been basically inactivated or closed up to not allow it to recognize. Impacting the viral RNA, the reverse transcriptase inhibitors to inactivate the RNA from being converted to DNA. The integrase to target after the DNA has been created, to target it and not allow it to move into the nucleus and become part of that chromosome, or after it has integrated, targeting the specific proteins, the protease inhibitors, not allowing it to package up as it should. So numerous potentials. And oftentimes with this, we see multi-drug approach where we've got a handful of classes of antivirals that are used simultaneously to try to impact this as much as possible. And there are definitely huge advances when we see treatments with HIV, bringing people down to undetectable levels within their body, which I think is awesome. We are learning more and more about this and ways to truly manage, which is definitely a benefit. So there are a handful of mechanisms that some organisms can utilize to try to evade those actions of the antibiotics, whatever they are, right? These bacteria or fungi, as we mentioned when we first started talking about this, when organisms evolved the ability to start producing some of these antimicrobial agents, there were other cells that started developing strategies to deal with those agents, like the beta-lactamase, that enzyme to chew it up. That's a pretty good example of a strategy to target specific things. And right here, we can see a handful of others for inactivation of enzymes targeting those specifics. So there's some where they potentially have a pump so that antimicrobial makes its way in, and with the efflux pump, it basically ships it right out. So it doesn't give that, whatever that agent is, it doesn't give it enough time to be inside of that cell and cause the damage that we're hoping it does. There's others that block the entry, so not allowing that specific class to come into the cell in the first place, or modifying as they try to do their actions, it targets it and does not allow for that to occur. And there's a couple of others that also impact it. Some of these organisms have the potential to carry some sort of resistance gene on a plasmid, and they can share that with some other microbe, some completely different type of microbe that now has the potential to pick up a type of resistance strategy that it never had before. And that's where we start thinking about the potential for some of these super pathogens that have evolved or picked up resistance, maybe handfuls of different types of resistance, lending to, well, serious concerns when we think about it. So there's ways, there's techniques that we can use to evaluate classes of antibiotics and see how effective they truly are. And we showed in the last chapter the plates, the Kirby-Bauer technique with the disc diffusion, where you've got a handful of antibiotic discs put onto this plate that's got the lawn of growth. You incubate it up and see which specific type of antimicrobial has the biggest zone of inhibition, killed the most, right? And that's one mechanism. It is really a kind of a qualitative type of a procedure. You've got a finite amount of antibiotic placed on into that, impregnated into that disc that's placed onto the plate. The image that we've got here is more of a quantitative. You can get a specific dilution. You can figure out what concentration of the antibiotic is going to be the most effective. And I want to stop right here and say, as we look at this, we're looking at, a specific amount of bacteria in this inoculum and a specific concentration of antimicrobial agent. The minimum inhibitory concentration, which is what we're seeing up here, is what will kill, it's the minimum amount of that drug that will kill the visible amount of bacteria, all right? So could there still maybe be some bacteria here? Well, it's not turbid, it's not cloudy like this, but maybe there are a few. And just because it's 8 micrograms per milliliter to kill this visible amount, maybe that's not uh, the dosage you truly want to prescribe to a patient, right? So there's other techniques when we really think about what's the dosage that you would be providing to a patient. This clues you into that, all right? So the MIC, it's the lowest dilution that inhibits a visible amount, all right, of organism. Like I said, there could potentially still be some in that, all right? So you can in turn see other stuff, broths without turbidity. So these guys right here, they could be inoculated onto plates that don't have antimicrobial drug. And now the lowest dilution that we see, killing 99.9% of that starting inoculum. That's this MBC, which is the minimum bactericidal concentration. So that gives you, kind of clues you in a little more as far as how much would truly be effective, all right? This, like I said, maybe there's a few that are still viable in here, all right? Usually, as you look at this, you've got a handful of dilutions, as we see here, and the antibiotic, the concentration increases as we go up, in this case by a twofold increase. And so at 8 micrograms of whatever antibiotic per milliliter we're talking about here, it appears to be effective, right? And plating it out, maybe we see that this concentration also shows no growth as it's been plated out. And that really would be the same, the MBC, all right? Okay. And it's typically more common to just talk about MIC, the minimum inhibitory concentration. So the lowest dilution that inhibits that. So it can occur in test tubes like we just saw. We can also do a susceptibility test on a microtiter plate. That's what we've got up here. This is a 96 well little plastic plate that you can do your dilutions in and visualize kind of the same sort of an idea whether or not this has occurred. So we've got from low to high concentration of antimicrobial agent. And in this case, we've got the potential to have a couple of different classes of antibiotics tested on the same plate. You could think of the previous slide, same sort of an idea. You just have racks of tubes with the specific amounts of bacteria and then different drugs diluted into each of them. Same sort of an idea. This is just a little easier to deal with, all in one plate. So again, higher concentration and lower on the left-hand side. So the minimum inhibitory concentration, as we're looking at this here, we've got, it looks like controls. So where you've got these bits are the minimums, right? So right here, where there should be, this is where it should be, all right? Where we're not seeing any turbidity. So this, here we've circled another, these are all where we're going. And up here, as we're looking at this, it's greater than this because we diluted it. This is as far as we've diluted it, and we're still seeing it. It's more than that, right? And maybe, as it turns out, this microbe is resistant to clindamycin. We've got growth in every single well, right? So it either means you need to continue to dilute, or this drug's just not effective, right? It's hard to say. And they're not telling us other antibiotics there, but you do see variation within it. This e-test is, it's almost like that Kirby-Bauer technique that we showed in the last chapter. But instead of it being a qualitative approach, this is more quantitative. It's kind of like you're doing the tube dilution, the minimum inhibitory concentration. It's kind of like you're doing that, but you're doing it on a petri dish with that lawn of growth, right? So this antibiotic is, in this case, we've got vancomycin, VA. So vancomycin is impregnated into this strip of paper, and each little number on here is the concentration of that drug. So it's like, again, that big tube test. Highly concentrated to very dilute. And what we can do is it's almost like it's pointing right to it as we look at this. So you can see very concentrated. We've inhibited a lot of the bacterial growth, and we keep moving in, and we get down here to the 1.5, in this case 1.5 micrograms per milliliter. That's the MIC, the minimum inhibitory concentration to inhibit growth. After that, it's all cloudy, right? And as we get even more dilute, it's, well, obvious that the bacteria grows right on it. You can see here at about the 4, there's a bunch of little bits of growth. So it's kind of a little more bacteriostatic, inhibiting or slowing the growth, but maybe not necessarily killing. But you can see the line kind of approaching right to this 1.5, and that's where you'd rule it out. It does, again, does not mean that that's the concentration you would prescribe to your patient. It's likely going to be, usually you find that minimum inhibitory concentration, and it's usually about 3 times that dosage that you would prescribe to the patient, right? Give or take a little bit with that specific. So, you know, antibiotics, when we think about the production, the discovery, all of that stuff, I'd like to say it's big business, but it's maybe not so much anymore. It's maybe not really that good of a business at all. Some of the pharmaceutical companies seem to think, and there's definitely issues with this. Oftentimes, a new class of antibiotic, or just a new antibiotic, doesn't have to be a new class, just a new antibiotic, once it's been approved and it starts to be utilized commercially, you only have a couple of years at best before you start seeing resistance to that specific drug. Doesn't mean that it's no longer useful, but there's definitely issues. And it kind of begs the question, why waste your time finding these agents when they're not going to be as useful? So lots of companies have kind of backed off on these ideas. It is expensive to find, to get it to market. It costs a tremendous amount of money. And to know that it's not going to have that long before there's issues causes them to kind of wonder, why waste their time? Approvals for new antimicrobials have declined considerably. And with that, it kind of makes you wonder, are we nearing the end of this antibiotic era in the first place, where there's very few that are being pushed to market? It's hard to say. That, you know, antibiotics are, you know, or the organisms to produce them are really kind of all over. It just, you know, you just take a little digging in the soil and you may be able to find some of these actinomycetes or maybe some fungi that harbor some skills that we have yet to understand or to discover. But again, with resistance, is it a good approach? Is it worth our time to continue through with that stuff? It's hard to say. So I do know, that there are some, newly discovered that may harbor true benefits, and there are others that are so harmful that you wouldn't want to use them on your body in the first place. So there's definitely some issues and concerns with that. Um, okay, let's see. Stop sharing this. Oh, this says now I'm streaming on YouTube. Is that true? I guess we will see. I tried. Okay, I think we've got just a few minutes early. I'm going to stop right now because I didn't get on this computer. I didn't get the other chapter downloaded. I'm not ready to start that one. I am, but I'm not. We will start with immunology stuff Wednesday. Like I said, I will post details about the extra credit paper soon, before the end of today. Any questions or concerns?
So will we have both the extra credit paper and an extra credit quiz that we need to be watching for?
Yes, I will. There will be extra credit paper. I know there were a few people with that very first extra credit that missed it, and I want to give you guys an opportunity to get some points if you did miss that. So this paper will be extra credit. There will be extra credit quiz stuff that is like practice test. I'll have that posted on there pretty soon as well. Any other questions? Okay, guys. Thank you. Have a good day. Oh, I've got
Thank you.
Thank you. See my chats. Oh, they're just thank yous. All right. Have a great day.